This webinar will cover BioPsych gene pages, operon pages, and search tools for gene and operon information. Let's start at the ecopsych.org homepage and begin by searching for the E. coli gene THRA. And I will simply hit enter to do the search. The search results are organized by object type. Here are search results against genes, against proteins, against metabolites, and there are more search results further down. We're basically seeing all objects that contain the string THRA somewhere in their primary name or their synonym. Let's click on THRA here to go to the gene page. The gene page is organized into three different sections. There's the upper section here that's fixed. This lower section shows information from one of multiple different information tabs that can be selected. And then we have this operations menu on the right. The upper area provides the gene name, synonyms, product name, and accession numbers, and reaction, pathway, and evidence code information for this gene. The lower area initially contains several diagrams and a mini review summary here authored by our curators. We created this regulation summary diagram to graphically summarize all known regulatory influences on the current gene, including regulation of transcription by PPGPP here, regulation of translation by isoleucine and threonine, and post-translational regulation by threonine. Clicking the green question mark will give more information on the workings of this diagram, the meaning of this diagram. Database links are also present in the Summary tab. This diagram shows the relationship between a gene, its protein product, a complex formed by that protein product, and one or more reactions catalyzed by that complex. And like most other diagrams, we can zoom this diagram by moving into it and either using the mouse wheel or using this zoom bar here. And we can also print the diagram using the printer icon. Now, to make it easier to find additional information about a given gene, we've organized additional information into these additional tabs along the top. The gene ontology tab is fairly straightforward. It shows gene ontology terms assigned to this gene. The terms are divided into several categories related to the biological process in which the gene is involved, the molecular function in which the gene is involved, and the cellular location of the gene product. And listed here is the gene ontology term itself and an evidence code that tells us is there experimental or computational evidence for this assignment and which database actually made the assignment. And you'll see that we've imported a lot of gene ontology information from UniProt. The Essentiality tab summarizes gene essentiality information available for this gene, noting whether knockouts for THRA grow under a variety of media and conditions. Here we see that a THRA knockout does grow under LB enriched medium, whereas for MOPS medium with 0.4% glucose, no growth was observed. The Reactions tab lists the reactions catalyzed by the gene product, in this case, two different reactions. Reaction 1, Reaction 2. Because an enzyme with multiple active sites may respond to different activators and inhibitors at each site, we list activators and inhibitors separately for each reaction, as well as and similarly the kinetic parameters may vary for the different active sites, so they're also listed separately. The Protein Features tab colors the protein sequence at residues of interest. This protein has many sequence features listed, many of them derived from UniProt. For example, at residues 471 to 478, there's a nucleotide phosphate binding region. That's up in this area. The Operons tab shows the operon containing this gene plus one gene on either side of the operon. Furthermore, for clarity, when there's more than one promoter, we draw a separate diagram for each transcription unit. A transcription unit is a group of genes controlled by one promoter. So this is the overall diagram here showing all promoters, one promoter here, one promoter here, and then we should draw different diagrams for the two different transcription units starting in each promoter. If the promoters are close together, it may be unclear which transcription factor binding sites control which promoters. By drawing them separately, we can make those relationships clearer. The References tab shows all references for this gene. The Show All tab combines information from all the tabs together to facilitate searching via your web browser. And the Modeling tab shows information from the E. coli whole cell model developed by Marcus Covert's group at Stanford. We're showing for 3 different growth conditions a number of parameters related to this gene that are calculated by the E. coli whole cell model. For example, the mRNA count for the THRA gene is shown under these 3 different growth conditions. And this graph at the bottom compares monomer counts predicted by the whole cell model against experimental monomer counts. And the points that deviate from the 45-degree line are areas where the model predictions deviate significantly from experimental observations. Let's now look at how to retrieve protein and nucleotide sequences for a given gene. Commands for those operations are found in the right sidebar Operations menu. In general, the commands in this right sidebar menu change depending on what type of page we're looking at. For example, they'll differ for gene pages versus metabolite pages. To obtain the amino acid sequence of the THRA gene product, we click Get Protein Sequence. So we've retrieved the amino acid sequence here in this web browser window, and this could be copied and pasted into another application or saved to a file. Let's go back to retrieve the nucleotide sequence of the gene. We'll click Get Nucleotide Sequence. Initially, this window is set up to retrieve the nucleotide sequence for the coding region. And so the requested region shown in this line contains the coordinates for the left end position and right end position of the coding region. And here's the requested length. Now we can modify that region to include, say, 100 upstream nucleotides. I'll enter 100 here and then simply click here and it changes the value. We could add, say, 500 nucleotides downstream, and we can say Show Sequence and close the window. And here's the full nucleotide sequence for that selected region, and notice that the start codon and the stop codon are shown in green and red. But the region you select does not have to be limited to being right around the coding region. It could be the entire chromosome if you wish. You can really enter, going back to this window, you can enter whatever upstream and downstream offsets you like, or simply change the left end position or right end position directly and click Reverse Complement to return the complement. Let's now look at the Gene Search tool, which is under the Search menu, Search Genes, Proteins, or RNAs. This tool is designed to enable construction of searches for genes that satisfy multiple criteria, as well as generation of lists of all entities of a given type. So for example, we can list all the genes or all the gene products or all the protein complexes in E. coli. Let's click List All Protein Complexes, and we get a table listing each complex in EcoCyc. And we can click on these to go to the protein complex page. And if we log in, we can turn this into a smart table, which is the topic of another tutorial. And let's go back, and we can also click List All the RNAs. And here we see all the RNAs, which can be sorted either by product name or by gene name. Let's do ascending sort by gene name, and there we have it. Those are simply listings of all entities of those types. Let's now look at how to do a multi-criterion search. Let's say we're interested in finding all gene products whose molecular weight is between 100 and 200 and whose PI is less than 10. Each of these lines gives a different filtering criterion for our search, and whichever criteria we provide are combined conjunctively. So for the molecular weight criterion, let's enter 100 and 200 as our upper and lower bounds. And then for PI's got to be less than or equal to 10. And let's do that search now. We'll submit our query here. And here's our result. So it gives us the gene name, product name, molecular weight, and PI. We could sort by PI, for example, and we see these are indeed all less than 10. We can sort by molecular weight, sort downward. And essentially, if I uncheck this box, then that criterion is now turned off for the search. Only this criterion would be active. We can look for cases where L-arginine is either a enzyme cofactor, inhibitor, activator, or transcription factor ligand. And here are our results. We can search for all proteins that have a map position in the first 1 megabase of the chromosome and have a sequence length less than 100 amino acids. And there's our search result. Another interesting kind of search that we can do is to search based on protein features information. Let's search for all proteins that have lipid binding sites, of which there seem to be 100. Let's submit that query. And so all of these, for each of these genes and products, we see gene name, product name, and what lipid binding sites they contain. And if we click on that site, it goes to the Features tab that shows where the lipid binding site is, which right here is shown at residue 25. And the lipids that bind there, there are 2 known lipids that bind there, which is right here. Let's now examine transcription unit pages. So back again to the THRA gene page, if we go to the Operon tab, if we click on one of the promoters here, that will take us to an Operon page, also called the transcription unit page. Same if we click on the Show Details button. So now we're on a transcription unit page, which shows both the view of this specific transcription unit and the larger view containing 1 gene on each side, as well as all promoters in this Operon. And as we scroll down, we see a summary written by our curators, and we see a section of this page for each of the sites in this transcription unit. So there's a section for the promoter that gives a citation for it, as well as evidence codes for it. So there are 3 different evidence codes. We see the sequence of the promoter region, as well as the exact start of transcription. And then we see a section for the regulation of RNA polymerase, and we also see under this growth condition, and we see the terminator listed, again with its sequence as well. Now I'm going to search for a different transcription unit page for ARGCBH. Here, I wanted you to note here that there are 2 transcription factor binding sites for ARGR, and they're numbered. There's site 1 and site 2, and if we scroll down, we see that each site is described with a separate section that identifies the evidence for the existence of this site, its sequence, citations, and the fact that it's ARGR bound to L-arginine that represses transcription. And thank you for your attention.